So we're discussing proteolytic enzymes. They're divided into two classifications. One is according to their site of action, and the other is according to their physiological focus, or another what part of the body are they primarily active in. So first of all, according to the site of action, whether they cleave internal peptide bonds or external or terminal peptide bonds. So internal or terminal. On the basis of that, they divide into endopeptidases and exopeptidases. Endopeptidases are pepsin, trypsin, chymotrypsin, and elastase. So pepsin, trypsin, chymotrypsin, and elastase. And exopeptidases, they cleave the terminal peptide bonds. And their examples are aminopeptidase and carboxypeptidase. Now, aminopeptidase, this removes amino acid from the N terminal. This carboxypeptidase, this removes amino acid from the C terminal. Now, when you talk about the other classification, which is according to their physiological focus, you will have gastric protease, which is pepsin, because this acts on only this one, this acts on the stomach, and trypsin, chymotrypsin, elastase, and also the carboxypeptidase. These four are basically pancreatic proteases. They basically act in the lumen of the small intestine. And then you have aminopeptidase, which acts on the brush border, according with the also in conjunction with the dipeptidase and tripeptidase. Alright, so let's look at this. First of all, pepsin, it is secreted as pepsinogen, which is in activated form so that it doesn't dissolve the stomach. Then you have, and this is activated, pepsinogen is activated by HCl, which is secreted by the chief cells. Then you have so this is very understood because obviously gastrics in stomach you have HCl, it's going to activate pepsinogen. Pepsinogen will be converted into pepsin, and then this further pepsin will activate all the remaining pepsinogen. And when you talk about pancreatic proteases, they are also secreted as inactivated zymogens. So trypsin, trypsinogen, chymotrypsin, chymotrypsinogen, elastase, proelastase, carboxypeptidase, and procarboxypeptidase. These are endo, this is also endo, this is exo, and this is also exo. Now these are activated by enterokinase, particularly this is activated by enterokinase, then the remaining are activated by the trypsin. So the trypsin is going to activate remaining of these. So these can also be activated by enterokinase. Then intestinal, you have these three. Now moving forward, it's important for you to understand that the gastric proteolytic enzyme in active form pepsinogen, which is from chief cells, which is activated by HCl. So pepsinogen is from the chief cells. Now this autocatalytically activates more pepsinogen. This is something that I've already clarified. Now pancreatic proteolytic enzymes, they are activated by, they're also treated as zymogens, and the key initiator is enterokinase. For example, trypsinogen is converted to trypsin. Then this trypsin is further going to activate all the remaining other pancreatic proteases. And chymotrypsin activated by trypsin, but it can also be activated by enterokinase. Elastase activated by trypsin, carboxypeptidase activated by trypsin. Aminopeptidase, dipeptidase, and tripeptidase, these all appear as the activated um, parts only, so they're already activated. Now this is the side stomach. You have pepsin. In the intestinal lumen, you have pancreatic proteases. Brush border, you have peptidases, and these are the products. And then when you just you need to know this one particularly, the pancreatic peptidases, they make oligopeptides and amino acids. And yes, so pepsin activated by HCl or pepsinogen, you can say rather trypsinogen activated by enterokinase, chymotrypsinogen, proelastase, procarboxypeptidase activated by trypsin, and aminopeptidase, dipeptidase, and tripeptidase, they're already active.